Hi everyone, and you're very welcome to today's Saturday session, where our focus for today is going to be Leaving Cert Biology, and specifically we're going to look on the topic of food. Now, before we get into looking at the topic of food today, I just want to give a quick overview of the layout of the exam, which is going to happen for 2024. It's similar to what has happened in 2022 and 2023. So for section A, we're going to have our short answer questions. As always, they're going to be 25%. There's going to be 7 questions. That used to be 6, that's been increased to 7. You're only required to answer only 5. Each one carries 20 marks, so that gives a total of 100 marks. Section B is the experiment questions. So there are 3 questions to answer in here. You only have to answer 2 of them. Each one carries 30 marks, so that gives you a total of 60 marks or 15%. And then the last ones are the long answer questions. So there's more choice within these now as well. There used to be 6 questions, there's now 7 questions. You're required to answer only 4 from 7, and each one carries 60 marks, so that would give you a total of 240 marks for the long questions. So all in all, the paper is going to be out of 400 marks. Okay, so the reason we're going to be looking at food today is because when we look at the structure of the exam, the structure of the course, we can see here that food actually falls within Unit 1. Okay, it's one of 3 topics in Unit 1, along with just the scientific method and ecology. Okay, so there's only 3 topics there. Move on to Unit 2, you can see there's a couple more, and then Unit 3 is by far the biggest unit that we have in the Leaving Cert Biology course. Now, this is actually really, really important for us, because if we think back to the slide we just seen there about how you have to answer 5 short questions, 2 of those short questions are going to come from Unit 1. So there's a real possibility that food will be one of those. And more often than not, it actually is one of the short questions that will pop up. So when we look at the course structure to the exam layout, this is useful for all topics, not just for food, which we're going to be looking at today. In the short answer questions, 2 of them will come from Unit 1, so food, scientific method, ecology. 2 of them will come from Unit 2, and 2 of them used to come from Unit 3. Now, we do have an additional 7th question. That 7th question can actually come from any of the units. Most likely would pop up there in Unit 3, because obviously it is the biggest one. But 2 of those short questions are coming from Unit 1, so there's a real chance that food will be one of those. And it's a very, very short topic. An experiment question could come up there as well. So the food tests, we actually do know which question this will pop up on now. I'll be doing this in a subsequent video. But the food test could come up as an experiment question. Okay, so there's a real chance that you're doubling up on marks there. And food does kind of interweave throughout other topics as well. So when we're talking about the cell, we're bringing in the phospholipids into the structure of the cell membrane and so on. So it's a really, really crucial topic, which underpins many other ones as well. So we're going to get into food for today. And the first thing we're going to start off with is the need for food. So nutrition is the way in which an organism will obtain and use its food. And nutrients are required for us in a couple of different ways, okay? So the nutrients we will be getting day to day in the food sources that we would eat will be things like minerals, carbohydrates, fats, we have vitamins, we have water, and we have proteins, okay? Why do we actually need to have food? Well, we need it for energy, we need it for growth and repair of our cells and muscles, and we also need food to make chemicals that are needed for cellular and metabolic reactions. Okay, and that is a very, very common exam question which has come up before, to outline or state 2 or 3 reasons why we might need food. Okay, and that's your reasons there. We need it for energy, we need it for growth and repair, and we need it for chemicals for cellular and metabolic reactions. Now we're going to look at the food components, okay? And throughout this, we're going to be focusing in on carbohydrates, we'll be focusing in on lipids, we'll be focusing in on proteins, and then we'll start to have a look at the vitamins in a little while as well. For now, though, we're going to focus on carbohydrates, lipids, and proteins. Now, carbohydrates, lipids, and proteins will generally be made up of a combination of these chemical elements over here. So these are the 6 main chemical elements which can be found in food, and it is crucial to know what they are, okay? So we have carbon, we have hydrogen, we have oxygen, phosphorus, we have sulfur, and we have nitrogen. Now, you're probably thinking, how am I going to remember which one of those chemical elements make up each of the different food biomolecules? You'll already know some of them. So an example of a carbohydrate, for example, would be glucose. We know the chemical formula for glucose is C6H12O6. So straight away you can see there that particular carbohydrate, and actually all carbohydrates, are made up of carbon, hydrogen, and oxygen. Okay, and we will go through each one of those in a couple of minutes, and which elements each biomolecule is made up of. But for now, really, really important, just remember CHOPS and carbon, hydrogen, oxygen, phosphorus, sulfur, and nitrogen. They're the 6 main chemical elements which are found in food. Now, we do have some salts which are found as well. So we have sodium, we've magnesium, we've chlorine, we've potassium, and we've calcium. And we do have a number of trace elements as well. Okay, so what this word trace means here, it's in very small quantities. So they do have a role inside our bodies, but we only need them in very, very small amounts. Okay, and an example of a trace element would be iron, we've copper, and we have zinc. Now, the biomolecules are what we're going to be focusing on over the next little while. So these elements, when joined together to form the 4 different main types of food biomolecules. We've actually already seen one of them there when we talked about glucose. That was an example of a carbohydrate. But for the next couple of minutes, we're going to be focusing on carbohydrates, lipids, proteins, and vitamins. And it is important for each one of those to know which of the elements which you've already seen, which of the salts and which of the trace elements you might actually be able to find within each of the biomolecules. And it's also important to know where you can actually find these biomolecules in different foods that you might be eating day to day. Okay, you do have to draw diagrams of certain amounts of them as well. So the carbohydrates and the lipids, for example, you do need to know what they actually look like and be able to draw a diagram of the individual components of them. But we will get to all of that in a couple of minutes. So the first one we're going to be looking at here is the carbohydrates. So of the 6 main elements we seen a couple of minutes ago, carbohydrates will contain the elements carbon, hydrogen, and oxygen. And again, if you're ever in doubt about which ones it actually contains, just remember that glucose is an example of a carbohydrate, and that's C6H12O6. You'll see that pop up again and again and again, especially in the photosynthesis chapter. But if you just remember that glucose is an example of a carbohydrate, and you know the chemical formula is C6H12O6, you can easily see that it's made of carbon, hydrogen, and oxygen. Now, this is actually really important for us down here when we look at the ratio that all of those elements are present within carbohydrates. It's always going to be in the form of CxH2OY. X and Y are generally always going to be equal to as well in that case. So the example of glucose being 6, if X was 6 and Y was 6, well, that would make sense because Cx will become C6. H2 will be multiplied by the 6 because we have 6 sets of this H2O. So 6 by 2 is 12, giving us our H12. And then O by 6 is O6, okay? So it's generally always in the form CxH2OY. And that is an exam question which does come up sometimes as well. It's come up in 2 ways. One way it's come up is it actually has given you this general formula here, and it's like, which of the main food biomolecules does this represent? And you would have to say the word carbohydrates, or it would say what is the general ratio or the general formula for finding the ratio of carbon, hydrogen, and oxygen in a carbohydrate, and you would have to just know that it's CxH2OY. Okay, and again, glucose, the example, C6H12O6. Now, carbohydrates are broken down into 3 different categories. We have our simplest category, then we go one level up, and then we go a little bit further again. So I just want to focus, I suppose, on the starting of each of these words here. We have monosaccharides, we have disaccharides, and we have polysaccharides. So I'm going to focus in here on mono, on di, and poly. Okay, the word mono means 1. The word di means 2, and poly means many. Okay, so if you think of glucose is an example of a carbohydrate. It's made up of sugar units. Okay, it's a simple sugar unit, 1 sugar unit. So a monosaccharide is a carbohydrate that consists of just 1 sugar unit. Okay, glucose being an example of that. By the time we get up here to a disaccharides are made up of 2 sugar units joined together. Polysaccharides then will be many sugar units joined together. Okay, and some examples of this. Glucose will be an example of a monosaccharide. That's just 1 sugar unit. Okay, maltose will be an example of 2 sugar units. Whereas starch and glycogen, they will be known as complex carbohydrates. The reason they're known as complex carbohydrates is because they're composed of many sugar units. Okay, they're examples of polysaccharides. So this is just a summary of what we've seen so far. We can see here we have a number of different monosaccharides over here. They're single sugar units, and we would know that they're very sweet to taste. Okay, if you've had glucose before, you know that it's very, very sweet, and all of those there will be soluble in water as well. Now, depending on what way you join them up to form your disaccharide, you could have 2 glucose units joined together. If you have 2 glucose units joined together, you'll form maltose. If you have a glucose unit and a fructose unit, you'll form sucrose. And if you have glucose and you have galactose, then you're going to form lactose. Don't worry too much about these examples here. Do focus though of how many sugar units we have. We have 2 sugar units in each one. That means it's a disaccharide. Okay, so we have 2 sugar units in a disaccharide. They're also all sweet to taste, and they're also soluble in water. Okay, If we have a look at our polysaccharides, we can see straight away these are all complex carbohydrates. Okay, starch up here formed of many glucose units, glycogen even more glucose units, fiber slightly less, but still it's composed of many sugar units, so that's why it's referred to as a polysaccharide. Okay, many sugar units, and all of these complex carbohydrates, or these polysaccharides, they're going to be insoluble in water, so it's the opposite from the monosaccharides and the disaccharides. Now, what are the function of carbohydrates? Why do we actually need to intake these as through a thread our diet or as part of our diet? So for each of these, for the carbohydrates, for the proteins, the lipids and the vitamins and so on, we're going to look at two, different types of functions. We're going to look at a structural function, and we're also going to look at it in terms of metabolism. So what's a structural function of carbohydrates and what's a metabolic function of carbohydrates? So a structural function, we're actually going to switch over to plant cells for this. Cellulose will form cell walls in plants. So that's an example of a carbohydrate, okay? And helps to form the cell walls in plants to give it that strength and that support and the protection that the plants will need. A metabolic function of carbohydrates is for energy and respiration. So glucose, also found not just in photosynthesis, but we actually use it as part of respiration as well. So that provides the energy. Glycogen will store energy in animals, and starch will store energy in plants, okay? So that's an example of a metabolic function. Now, where do we actually find these carbohydrates day to day if we wanted to make sure we are taking them in as part of our diet? Well, bread, potatoes, rice, pasta, sugars, fruits, sweets and cakes, all of those will be good sources of carbohydrates, and it is important that you know that as well. Now we're going to switch over here to our lipids. So these are our fats and our oils, and that is a question which has come up before actually on the Leaving Cert exam, is to give a difference between fats and oils. Both examples of lipids, but what you could say for that there are that fats are solid at room temperature. Not just enough to say that if it did come up as a question, you would have to also say that oils are liquid at room temperature. So very similar to carbohydrates in the elements that they're composed of carbon, hydrogen and oxygen. The ratios are not the same as carbohydrates. You're not required to know the ratio in this case, but it's not going to follow the one in carbohydrates where it was like CxH2OY. It's a different ratio. We've mentioned there already about fats being solid at room temperature, oils being liquid at room temperature. And there's two types you need to know here. And for this one, it is important that you actually know the diagram for this one. We have a triglyceride, just focus there on the word tri, that will mean 3, and that will make sense in a couple of minutes. And then we have our phospholipid as well. Okay, sources of lipids, we've our butter, we've our oils, we've cream, fried food. Any of those will be good examples of where you could intake lipids as part of your diet. Now for the triglyceride, tri meaning 3, okay, it's made up of one glycerol. So glycerol here is the backbone that we have, and it's composed of 3 fatty acids joined onto that. Okay, that's where the word tri comes from. Okay, tri meaning 3. So we have 3 fatty acids, and this is the smallest lipid. Okay, when we look at the phospholipid in a couple of minutes, it's going to be a little bit more complex. But for the triglyceride, we have our glycerol and we have fatty acid 1, 2 and 3. And hugely important that we know that diagram because it has come up in the exam before that you're asked to actually recreate that. The second type here, we have our phospholipid. So again, if we look at the start of this one, we can see we have phospho. That just refers to phosphate. So that means we're going to have a phosphate as part of our structure here. And we can see that straight away down in the diagram. Now it is very similar to the diagram we've seen in the last slide for the triglyceride. We still have our glycerol backbone there. We've 2 fatty acids, we fatty acid 1 and we fatty acid 2, but the third fatty acid has actually been replaced by a phosphate instead. So that's where the name phospholipid is coming from. And this is a hugely important structure of the cell membrane. So the cell membrane is composed of a phospholipid double membrane. Okay, so it would actually have 2 layers of phospholipids there. So when we get to a couple of minutes having a look at the structural and the metabolic roles of lipids, one really important structural role of those lipids is that it is a component of cell membranes. So carbohydrates are really important in the cell walls in plants. Phospholipids are really important in the cell membrane. So we'll see this now here anyway. So our lipids, we have our structural role, so it can store energy, heat insulation as well in our skin, protects our vital organs inside our bodies, and then phospholipids as well as proteins. There's actually 2 things within the cell membrane can join forming lipoproteins, and they're important structure in cell membranes. A metabolic role, lipids contain massive amounts of energy, so they can be broken down in respiration to release energy, which then can be supplied to all of the cells in our body. And just to focus in there on the cell membrane, we can see here, these are our lipids, okay? So we have our hydrophilic head and our hydrophobic tail. These two things here are our fatty acids, but we can see here we have a phospholipid bilayer, and that there is what's going to form the cell membrane. Now, when we get on to proteins, somewhat different from the carbohydrates and the lipids we've already seen. They do contain carbon, they do contain hydrogen, and they do contain oxygen, the exact same as what we've seen with the carbohydrates and the lipids, but they always contain nitrogen as well. Okay, sometimes they will also contain sulfur and phosphorus. Okay, so that's where those other three chemical elements we would have seen at the start come in. They come into proteins. Nitrogen will always be there, but sometimes sulfur and phosphorus will be there as well. Where can you find these proteins? Well, lean meat, fish, soya, and eggs. And proteins are large and complex molecules. Okay, their structure and their role is completely dependent on their amino acid sequence. There's 20, and that's an exam question which has come up before as well. There's 20 common amino acids that are found in the human body. So depending on what sequence they are in and what way they're folded, because it's this folding here that will give a protein its shape, their role will be dependent on that. And we'll see a couple of examples of that now in a minute. So this is the protein structure, and this is where their role and their function comes from. So proteins are composed of many amino acids. Okay, so the amino acids down here, they're the building blocks. So like the monosaccharides were the building blocks of the polysaccharides, the monosaccharide was the simplest unit. The amino acid is the simplest unit of a protein. Okay, so we can see we've many different amino acids completely separated there, but they're the simplest unit. Now, if we join or start to join them together, we would have what's called a peptide chain. Okay, that's generally if we have less than 20 amino acids. If we get to more than 20 amino acids, that would be a polypeptide chain. So it'll be many different peptide chains joined together. And then a protein will be even more complex than that. You would generally have to get in and around over 200 amino acids joined together to give a protein. As we said, they're very, very complex, so we're going to have lots of different amino acids in there. Okay, the amino acids are joined together by peptide bonds, and it's depending on the sequence in here and the folding, what shape this takes when it's folded. Because remember, sometimes there isn't an awful lot of space for these amino acids and these proteins to exist, so they will be folded in a very particular way, and that folding will dictate its function. So we have two types of proteins. We have fibrous proteins and we have globular proteins. So we can see here the globular proteins generally are more compact together. They're more like a ball shape compared to the fibrous protein, which seem to exist more in these strands. So the fibrous proteins will have little or no folding. They're long fibers, they're strong, and they're tough. And an example of this, so always an exam question as well, to give an example of a fibrous protein, will be keratin, which you can find in your hair, your skin, and your nails. That's why you might see some nail products and some hair products will say that there's extra keratin in it, or it's infused with keratin, because that's the structural protein that you actually find in your hair, your skin, and your nails. The globular protein, the reason it looks like this is because it has a lot more folding going on, and they form more rounder shapes, and enzymes will be an example of a globular protein. So if you were asked to give an example of a globular protein, you could say enzymes, but you could go one step further and give a specific example of an enzyme. So for example, you could give amylase or DNA ligase or RNA polymerase, something like that. So any enzyme would be a sufficient example for globular proteins. Now, where do we actually get our proteins? Well, we get them from meat, fish, eggs, nuts, milk, and so on. So they're all good sources of protein in your diet day to day. Now, what is the role of protein? So again, we're going to look at a structural role and we're going to look at a metabolic role. So structural role of protein, we've already seen. Keratin, found in your hair, your skin, and your nails. And myosin is another example actually of a protein, and that's found in your muscle. So it gives muscle structure. A metabolic role, enzymes are proteins. Okay, enzymes are protein catalysts or biological catalysts. So they're used as enzymes to control reactions. Okay, another example of proteins being used in a metabolic role will be to form antibodies to fight infection. Okay, so 4 different examples there: structural roles and metabolic roles. So that was our carbohydrates, our lipids, and our proteins. We're now going to have a look at our vitamins, and we're actually going to break these down into 2 different groups as well. So vitamins are complex carbon-based structures. Okay, so they're based off of carbon, they contain carbon, We only need them in small amounts throughout our diets because our bodies can't make them themselves, okay? They can actually, our bodies can't make them, but they can actually be formed in our bodies. So an example of that would be some bacteria can actually produce some vitamins in our intestines, but our bodies themselves can't actually produce them. Now, vitamins can be broken down into two separate groups, or a question which might come up would be, in what way are vitamins normally separated from each other or grouped apart from each other? It will be whether they're water soluble or whether they're fat soluble. So is the vitamin soluble, can it dissolve in water, or is the vitamin soluble in fat, or can it dissolve in fat? They can be divided up into both of those groups, and we look at two examples today. We're going to look at vitamin C and we're going to look at vitamin D. Vitamin C being a water soluble vitamin and vitamin D being a fat soluble vitamin. So the key vitamins which you might have heard of before, we have vitamin A, vitamin B, which is broken down into the B group vitamins. So you have B1 up to B12, you have vitamin C, you have vitamin D, you have vitamin E, and we have vitamin K. We're going to be focusing on these two here in the centre today, which is vitamin C and vitamin D, because both of them actually fall into one of these two groups up here, water soluble and fat soluble. So a water soluble vitamin. Some of my students would always ask, how do you remember which one of C and D is water soluble and which one is fat soluble? The way I would always remember it is, if you think of Berocca, a Berocca tablet is dissolvable in water, and that's a way to get extra vitamin C into your body, okay? So when you're taking your Berocca, you're dissolving that in water, okay? So that's an example of a water soluble vitamin. Some sources of vitamin C, vegetables and citrus fruits like oranges, lemons, limes, all really good sources of getting your vitamin C. Why do we actually need vitamin C? It's actually responsible for the formation of connective tissue. So we'll come back to the idea of the gums in a couple of minutes, but your skin, your gums and your cartilage, vitamin C is responsible for that. Really, really important for the growth and maintenance of bones and teeth. And it's actually also responsible for the aiding of the healing of wounds, okay? So all of this is vitamin C. Now, for both of these vitamins, which we're going to look at, C and D, not only do we need to know the source and whether they fall into the group of water soluble and fat soluble, we do need to know a deficiency associated with it as well. So what would happen in the human body if you weren't getting enough of this particular vitamin? And I should just say as well, it's very, very difficult to not get enough of these vitamins because we do only need them in really, really small amounts. So it would be very, very rare that this deficiency would have been seen. Would have been seen years and years ago in pirates an awful lot, but nowadays not so much. So a deficiency is a lack or a shortage of something. So a lack of vitamin C can cause scurvy. Okay, we seen a couple of minutes ago that one of the functions of vitamin C was it was responsible for the formation of connective tissue, but in particular it was responsible for the formation of the connective tissue within the gums. So if we're not getting enough vitamin C, our gums are going to be affected. So symptoms of scurvy, if you weren't getting enough vitamin C, will be poor healing of your skin. Again, vitamin C responsible for healing wounds, bleeding gums and loose teeth. Okay, coming back to that connective tissue not being properly formed, or the vitamin C we're not getting enough of it to help form that tissue. A fat soluble vitamin then, not vitamin C, this is going to be the opposite then. This is vitamin D, also known as calciferol. Some sources of this will be cod liver oil, milk and the egg yolk. Necessary for, it helps to absorb calcium from the intestine, and it's also responsible for healthy bone and teeth formation as well. So a deficiency of vitamin D, again, a deficiency being a lack or a shortage of something. It's actually called one of two things, whether this deficiency is found in children or adults. And there's a good description of what this actually looks like in a couple of minutes ago, which I will get to. But in children, a deficiency of vitamin D is known as rickets, and in adults, it's known as osteomalacia. Okay, so symptoms will be weak, deformed bones that break easily. So you can see here in the children who have rickets, their legs kind of bend outwards. Okay, their bones just aren't quite as strong. They're weaker, so they're a little bit more flexible than they should be. And the reason this happens, both in children and in adults, is to do with the size of the pores that exist inside the bone. So in a normal bone, if you have a look at the pores, they're generally quite small. But in a bone that might have osteomalacia or osteoporosis, the pores here are quite large. And the easiest way I get my students to remember this would be the inside of a normal bone would resemble what a crunchy bar would look like if you were to slice down right through that. There is pores there, but they're tiny. Okay, and if you try and remember if you were ever trying to break a crunchy bar in half, it can be quite difficult. Okay, that's because everything is so tightly compact and the pores are so small. Whereas if we have a look at the bone with osteomalacia or osteoporosis, the pores are much, much larger. Okay, and I would say to my students that would resemble what an aero bar would look like. Okay, the pores are there and they're much larger compared to, say, the crunchy bar. And again, the aero bar is much more easier to break because of the size of the bar, the pores that are in here. Okay, so that's a nice little way to remember the difference between both of those. So again, symptoms: weak, deformed bones, and they can break very, very easily. So that's all coming from a deficiency or a lack of vitamin D. So minerals are the last thing we have to look at here. So minerals are needed by both plants and animals in very, very small amounts. Minerals will include calcium, magnesium, and iron. And some of the uses for these minerals would include forming rigid body structures like your bones. So calcium, for example, there was an ad years ago saying them bones need calcium. So it's forming rigid body structures. Also can be used for your teeth as well. It makes soft body parts such as muscle, and then it can form salt in cell and body fluids as well. So just an overview of a couple of examples. You are expected to know examples for both plants and animals of what minerals are needed and where they're needed. So example, in plants, they need both magnesium and calcium. They would get both of them from the soil as they're taking in water. The magnesium will be used to form chlorophyll. Okay, so chlorophyll is the green dye that you find inside your chloroplast. Really, really important then for photosynthesis because it's going to absorb the sunlight for photosynthesis. Calcium is going to be used to help the cell walls attach to each other. And as we know, if you're to examine the plant cells underneath a microscope, they're very, very tightly compact together. And that's what gives the plants its rigid structure and helps to give it strength and support. Animals, they would need iron and calcium. Iron, where would you get this from in your diet? Well, liver, meat, vegetables. Iron then is responsible for forming hemoglobin. And that hemoglobin is responsible for carrying the oxygen around your blood. Okay, so the hemoglobin does that. And then calcium, we would pick that up from milk, cheese, and dairy. And again, we've kind of mentioned this already. That's responsible for the healthy formation of your bones and your teeth. Okay, now water. Obviously, we need to take in about 2 litres of water a day. And not only are we really dependent on water, plants are also very dependent on water as well. Okay, it's the most abundant chemical contained in living things. Could be up to about 90% or more of some cells. It's a component of the cytoplasm and body fluids, and it's a really, really good solvent. Okay, so it dissolves a range of molecules. It's also responsible for movement through membranes, so water can pass in and out of membranes very easily. And it's also a really good absorber of heat. So it's very slow to heat up and it's slow to cool down. Okay, and oceans being a really, really good example of that. Now, this is an exam question which has come up before based on all of what we've seen so far. Okay, taking into account most of the biomolecules we've seen. Now, the year this came up, it did say to just answer any 5 of the following parts, so there was a little bit of choice in there, but we're going to answer all of the parts here now. So, name the 4 chemical elements found in all proteins. Well, we know that the chemical elements which this possibly could be will be C, H, O, N, P, and S. Okay, so the 4 elements found in all proteins are carbon, hydrogen, we have oxygen there as well. They're also the ones found in carbohydrates and lipids, but we also have nitrogen in there as well. Okay, they're always present in proteins, and then phosphorus and sulfur sometimes are found. Name an element that is only sometimes found in proteins. I've actually answered that already there. It can be one of these 2 here, so it's either going to be phosphorus or sulfur. Remember, only give 1 answer when you're answering this as well, because if you do give 2, 1 is correct and 1 is wrong, the wrong answer will cancel out the correct answer. So only give 1 answer for a question like that. So sulfur, you could have said phosphorus as well. How many common amino acids are found in proteins? There's 20 common amino acids. Fibrous proteins have structural roles in living things or in living organisms. Name any one fibrous protein. So the fibrous ones, the globular ones, we said globular enzymes examples would have been enzymes. Globular proteins examples are enzymes. The fibrous protein, the example there that we had seen was keratin, found in your hair, your skin, and your nails. You could have said myosin there as well if you wanted to. Give 1 metabolic role of proteins. So how are proteins involved in metabolism in living things? Well, they act as catalysts or enzymes. And give 1 good source of protein. You can just say something like meat, okay, or fish, or chicken, or nuts, or eggs, anything along those lines. That would have been fine. So you actually only had to answer 5 of those parts there, so you could have left 1 of them out. But it was kind of taken into account a lot of what we had seen so far on all the different food biomolecules. So another example here, and here's the exam question I was talking about a couple of minutes ago. Identify 1 difference between fats and oils at room temperature. So fats are solid at room temperature. And make sure, don't stop there, you have to give the other side of the story as well. So oils are liquid at room temperature. This question did say to answer any 5 parts as well, but we're going to answer them all. Identify one difference between a triglyceride and a phospholipid. So tri and phospho. So a triglyceride, we can say has 3 fatty acids. And then you can say the phospholipid has 2 fatty acids and 1 phosphate. Okay, both of them do have glycerol as well, but it asks for the difference there. Okay, so identify one metabolic role for lipids in cells. So an example of this will be, it's an energy source. They can be broken down for energy. Name one fat-soluble vitamin present in food. So remember, the water-soluble one was the vitamin C. The fat-soluble one was vitamin D. Okay. Name one disorder associated with a deficiency of the vitamin named at D above. So we could say here that's rickets. You could have said osteoporosis or osteomalacia as well. And then we do have here, name one element present in dissolved salts in food. So the easiest one I remember there is either to put down sodium or chlorine, because if you remember that NaCl is sodium chloride, that's just table salt. Either the Na or the Cl will be accepted there in dissolved salts. So I'm just going to put down Na for sodium. And I'm actually just going to put down in brackets there after the actual word as well, because it did say name. So I want to make sure I get the marks there. So just put down the word sodium as well. Okay, so another exam question then based on the different food types. So here's the third exam question. So state 2 main reasons why food is required by all living things. We've seen this already. So it's an energy source. And also we have the growth and repair of cells. Okay, what is the ratio of hydrogen atoms to oxygen atoms in a carbohydrate? So if we have a look, remember carbohydrates, the example we had a couple of minutes ago was C6H12O6. Okay, that was the example. Okay, that was glucose. Have a look at the ratio between hydrogen to oxygen. So there's hydrogen is 12. Oxygen is 6. So what is the ratio? Well, you could write down the ratio is 12 is to 6, but always give it in its simplest form. So divide everything there by 6 and you get 2 is to 1. And remember, don't do 1 is to 2. Definitely don't do that, because it did ask hydrogen to oxygen. So hydrogen has to come first. So 2 is to 1. So give a structural role of lipids in cells. So where do we find lipids, or how are they responsible for structure in either plant or animal cells? Well, they're a component of cell membranes. A metabolic role of lipids, again, they're an energy source. We seen that question there a minute ago. Give an example of a fat-soluble vitamin. Again, that was vitamin D. And name a disorder, that's going to be rickets. So you can see there's some repetition in the questions there already, and that was only going back over the last 3 or 4 years of questions. Now, just one difference there, just to mention, I know it didn't ask it, but if it did ask for a water-soluble vitamin there, we would have had to say vitamin C, and then we would have just changed that answer there to scurvy instead. So the other thing that you need to know as part of the food chapter is the different food tests. So there's 4 different food tests that you would perform. You probably usually would have done these at the start of 5th year, and they can come up as a short answer question, or they could come up as part of an experiment question. Generally, you wouldn't have a full experiment question based off of these, but it is important to know them. And just the main thing about these is, what chemicals did you use and what colour did the chemical change if the particular food you were testing for was present. So you need to be able to test for reducing sugar, glucose. You need to be able to test for starch. You need to be able to test for protein, and you need to be able to test for lipids. Now, in each of the tests we're about to do here, we're going to set up the test using the food source that we're fairly sure will give a positive test. So we're going to be using glucose, because we know glucose is a reducing sugar. We also have to set up control for comparison as well. Water is generally a really, really good control to use when we're looking at the food tests, and we'll get to that as we look at the intricacies of each test in the next couple of minutes. So we're going to add glucose to water in a test tube. We can see that over here. So we have glucose in water, and then this one here is just going to be water, okay? Making sure we have the same volume in each test tube. Now, the chemical that we're going to be using for the test for the reducing sugar is Benedict's solution. Okay, and there's a particular colour I'm highlighting, or a particular reason why I'm highlighting this in red, which I'll get to in a couple of minutes. So in one test tube, we have our glucose solution and we have our Benedict's reagent. The other one, we just have water and our Benedict's reagent. Okay, we will place both of these test tubes over a heat source. We will heat it, we will not boil it. Okay, if the reducing sugar is present, the solution should turn brick red. Okay, so in this test tube up here, which has the glucose solution, that should turn brick red if glucose is present. In the one with the water over here, that's just going to remain blue, and I'll actually just colour that in there so you can see that as well. Benedict's reagent is blue, so that should turn blue there, and this one over here will change to brick red. Okay. The test for starch then, we're going to set up 2 test tubes the exact same way. Our control is going to be water, and we're going to add a few drops of iodine to a starch solution. Okay, so iodine is kind of an orangey brown colour. So when we add it into the water, that's the colour the water should turn. Starch solution, if iodine is present, the colour that we will have is blue-black. Okay, so it's a very specific colour. We have to term it blue-black down here. So that should be the colour that we get in the starch solution, blue-black. The test for protein. So we dissolve a soluble protein such as egg white in water. Okay, the egg white will dissolve straight away. So you could add that to a test tube with water. Water is just going to be the control that we use then as well. So there's 2 different options that you can have here. One is you can add sodium hydroxide until it remains clear into both of the test tubes, and then add drops of copper sulfate, which is a blue colour. Or if you add burette reagent or burette solution, that actually does both jobs for you at the same time. And that generally is probably the one that you would use in the school lab, but you can use either or. So use the sodium hydroxide and then add the copper sulfate, or just add burette solution straight away. Okay, if the colour changes to purple, protein is present. Okay, so this one here, because we have the egg white in there, that one should change to purple. This one over here should stay as blue, okay, because there was no protein present in that test tube. And test for lipids. So again, we're going to get 2 pieces of brown paper for this one, so you don't actually use test tubes at all here. On one of them, you're going to rub a piece of butter on the test piece, and the other one, you're just going to rub a piece of water or get some water and rub that onto the control piece. Leave both of them to dry. The one with water should look the exact same, might just be a little bit more crinkles because it's after been getting wet. But there should be a translucent mark, not transparent, a translucent mark. So if you hold it up to the light, you should start to be able to see through it, or a permanent stain will appear. That means that fats are present. Okay, so that's the test for lipids. Now, this table here is really, really useful for comparing all of the different food tests. So it might be useful to screenshot this because this is generally what the exam questions will be based off of when they pop up. So the test for reducing sugar, you were testing for glucose, you used Benedict's solution, which is blue. If it was absent, like in water, it will stay blue. But if after heating glucose with Benedict's solution in it, it should turn to brick red. Starch, an example of that would have been food. So using iodine, which is the orange-yellow colour, if it's absent, like it is in water, it will stay as that orangey-yellow colour. But if it's present, it will turn to blue-black. Lipid, an example of that would be butter. We use our brown paper for this. There will be no stain if you use something like water, but there will be a translucent mark or a permanent stain if it's present, like in butter. And then for protein, we actually used egg white dissolved in water. We used burette solution. It will stay blue in water because there's no protein there, but that blue colour will change to purple in egg white, or even in, say, protein milk, something like that, because protein was present. So one final exam question here. In carbohydrates, which 2 elements are in the ratio 2:1? We've seen a question very like this a couple of minutes ago. That was our hydrogen and our oxygen. Cellulose is a polysaccharide. Explain the term polysaccharide. So that's a carbohydrate composed of many sugar units. Name a polysaccharide other than cellulose. So starch will be an example of that. Where precisely in a plant cell would you expect to find cellulose? That's in the cell wall. That was part of the structural role of carbohydrates. Name a test or give the chemicals used to demonstrate the presence of a reducing sugar. That was Benedict's solution. And that would turn brick red if the reducing sugar was present. And in relation to the test referred in C, which of the following is correct? Is there no heat needed? Do you heat but not boil? Or do you boil? And that one there is heat but do not boil. So just before we finish up, I just want to take you through the ExamRevision website where you can access loads more content exactly like what we've been going through today. And you can find tutorial videos based not just on food like we were talking about today, but all other topics on the Leaving Cert Biology course. So when you log on to ExamRevision, you can do a month-by-month subscription if you like as well. When you log in, you'll see that Leaving Cert Biology there. We have our video tutorials, we have our presentations, we have our exam builder, we have quizzes, and we have our notes. So if we click into video tutorials, we were looking today at Unit 1, the study of life, and we were looking at nutrition or food. So you can see there that we have a video on the biomolecules, their structure and function, and on the food tests. Okay, exactly like what we looked at today. So you could watch back over that just to refresh your memory. When you're happy with that, you can press on the quiz over here, and we can see that there's a 10-question quiz based on everything you need to know about food. You will get your score right at the end once you have that completed. And when you're happy that you know the quiz, you can go right down here to the bottom and build your exam. So the exam questions which we would have had a look at today are already there. So you can make your own exam by going to the bottom of the questions, pressing the plus button on whatever questions and whatever combination of questions you want. You can choose any other unit as well. It doesn't just have to be food. And when you're ready, you just download the questions and the answers and you can test your knowledge. We do have a pack of H1 notes as well. So you can download these, print them off, and you will be able to see all of the content, all of the information that you need to know for that final exam. Okay, not only do we have Leaving Cert Biology, we have a wide range of subjects available on the platform, and it is cheaper. The more subjects you sign up for, the cheaper it is for you as well. So make sure and check out the ExamRevision website for all you need for your Leaving Cert subjects. There will be lots more Saturday sessions coming up over the next couple of weeks, and I hope to see you again for Biology 1 soon.